Ice,
Yep.
what do you think? What time do we start? This is actually 7 minutes after the scheduled time. Do it until 10 minutes after.
Yeah,
like
I think
3 more minutes.
maybe like let's wait for like maybe 2 or 3 minutes, and then we should just start. We have a pretty good amount of people here, so
yeah, so you can begin at 10 past, is it?
yeah, like
That's okay.
so while we're waiting, one note from the previous lecture. So we have the recording for all of our lecture, but we are double checking that the video quality is okay, and we are trimming down like this kind of waiting time from the video, just to make sure that you don't just like spend 10 minutes looking for nothing. So the video will be shared soon after Haiti, like the previous instructor from the first class, has take a look at it. Then we will be able to share the video with you guys, so that you can take a look, just in case you missed part of it or you want to double check what is going on. And then again, the homework will be graded soon after this class. But if you haven't worked on it yet, feel free to work whenever like you have time, and we will grade it like, let's say maybe up to once a week, or like whenever like our time is permit. And then after you get some feedback, it's okay to like keep talking in our in our Discord if there is any further question. So if any of you are still not part of the Discord, please double check it in the email, because there'll be like an invitation link to get into our Discord community, and that will allow you to ask like several questions related to the class, related to the homework, or maybe that would be the channel that you can connect with each other. So I think like we're getting into the time that we can start, I guess. So maybe Steve, you can like get started. Sounds good. Cool. Good evening, everyone. Let me introduce Steve a bit. So Steve is gonna be our second instructor for us today, and the talk today is gonna be about the central dogma, or like if you take it in a simple term, it's gonna be like how biology process the information in the system. So the lecture today will be very important for your understanding about biology and then synthetic biology in general. So if you have any question, please feel free to put into the chat, and then I can help answer it. Or if you have any further question after the class, please feel free to put into the Discord. So take it away, Steve. Thank you.
Thanks, Ice. I hope I'm audible enough, and I hope you can all see the screen. Confirm the audibility, Ice, with me. Is it? Can you hear me?
Yeah, I can hear you, and the screen is now like full screen. Looking good.
Okay, thanks so much. Great stuff. So, as I said, I'm Steve Melamia from Malawi, and I'll be taking you through the second lecture within this program of the 6-week training that we have. And as I said it, this comes to form a basis from what we had last time. Those of us that were able to join from our last lecture, where we had Haiti, who gave a lecture on what synthetic biology is all about. And we do understand that for us to dive deep into synthetic biology, the knowledge of molecular biology, specifically the central dogma of molecular biology, remains crucial, remains key. And it was necessary for the team to have this chipped in, so that we know what molecular biology is all about, specifically the central dogma of molecular biology, so that as we move forward with our growing interest in synthetic biology, we know and we are very much informed of how much we can do with the knowledge that we got last time in the synthetic biology. So feel free to ask questions. Probably we'll have time maybe at the very end where you can ask questions and yeah, help each other where you see clarification or the areas that you didn't get them well. Just a moment. Okay. So when you talk about central dogma of molecular biology, this is something that is very wide. For our friends that are coming from a biological background, you should have an idea what the central dogma of molecular biology is all about. For those that are coming from non-biology background, I'll make sure that I break it down for you in a simplified manner so that you know what this is all about and so that we can move together. We know this all these courses have been made in a way that blends in those that are coming from biological background and also our friends that are coming from non-biological background. So we will try to strike a very good balance that by the end of the day, each one of us benefits that we meet, halfway. So basically, when you talk about central dogma of molecular biology, we are talking about how the genetic information flow within a biological system. So primarily, it involves the three major components. We'll have the DNA. We're going to see what that means, the RNA, and then proteins. And this was first proposed by Francis Crick in 1957, and later it got elaborated in 1970. It is a fundamental concept in molecular biology as it explains how genetic information is stored, transferred, and most importantly, how it is expressed in all living organisms. I think this is very key when you talk about the expression. We are going to see how this plays a role when we talk about, in the arena of synthetic biology as a discipline on its own. So we do have what we call the unit of inheritance. Now, when you talk about the unit of inheritance, I want to break it down in this way. I know some of us we've heard perhaps from our friends, maybe colleagues, that we might have taken after our parents, maybe the way we look, our appearance, maybe we resemble our parents. Or if you have kids, most of us, maybe our kids might have taken after us, which simply means they might have inherited some traits from us that make them resemble us. So in simple terms, we're talking about the substance that defines you and me. If we say you look or you've taken after your mom, what are we saying? So in biology, we have these what we call unit of inheritance, and basically these are what we call nucleic acids. So they're contained in a chromosome, obviously in the cell, in the nucleus to be more specific. And then we do have two types of these nucleic acids. We've got what we call the DNA, which is in full deoxyribonucleic acid. And then we've got RNA, which simply means ribonucleic acid. I think you're going to see how these two differ in a number of ways. But I think one thing to note on this slide is that so these nucleic acids like DNA molecule or RNA molecule, they've got segments within themselves that code for proteins. And these segments are the ones that we call genes. And then, so these genes, these segments, like both DNA and RNA molecules, have got these different kinds of genes. I think we just have to take note, and then I will explain what more on the same in the coming slides. So we just want to have a look at DNA structure and the arrangements in the shortest time possible. So when talk about the DNA, this is basically a sort, and it's got these three major features. Number one, these are very important things to note. It has got a nitrogenous base. I don't know if my pointer my pointer is visible on that side. So this is the nitrogenous base. It's a heterocyclic ring composed of carbon and nitrogen atoms. And then it's got a 5-carbon sugar in a ring form in the name of pentose. And lastly, it's got a phosphate group. So when you talk of DNA, we are looking at these features. But also it's got what we call the nucleotides, the 4 nucleotides in the name of in the letters A, C, G, T. These are also called bases. And then you're going to see in the next slide what these bases stand for, these letters rather. So the G stands for guanine, you've got A, which stands for adenine, you've got T, that stands for thymine, and the last one, C, stands for cytosine. And then there are some cases where you just have letter R, and that would stand for either adenine or guanine, which are categorized as purines, or Y, which would simply stand for either cytosine or thymine, which are simply categorized as pyrimidines. Or when you have N could just simply mean N of those 4 bases. Where you've got an N, it simply means N of those 4 bases. So I think this also makes sense when you start looking at the sequences, the DNA sequences, RNA sequences in the subsequent slides. So the other thing to note here is that for RNA here, I've talked about the T here. So I'll talk about the first difference. In RNA, we do not have a T, a thymine rather. Instead, we have a uracil. So DNA, when you're doing DNA, you have GATC, but the case with RNA is a bit different, where instead of a T here, you've got a new. So I think something to take note, so that when we start dealing these things at a deeper level, it should be easier for us to flow. So I just want to introduce to you the model of DNA that was discovered by Watson Crick. That was 1953. So as you can see, it's a double helix structure. Number one, it's got the sugar phosphate backbone. I think, as I said in the last slide, but also it's got the nucleotide composition. Now, when we talk about the nucleotide composition, I think we need to get straight to our minds that these pair in the DNA structure. And then the mode of pairing, it's more of a complementary kind of pairing. So one thing to note on this slide is that A will always pair with T, that's in a DNA, and then G, which is guanine, will always pair with cytosine. And these pairing are supported or held together by hydrogen bonds. We're going to see in the coming slides what these kind of bonds are. So when you zoom this DNA structure, this is what we see at a deeper level, where we have the pairing a bit visible than what we had. So if you can see here, my pointer, we've got the first thing to note, the structure of adenine, and then you look at the structure of guanine, pretty much the same, close. And then you see the structure of thymine and then cytosine. You can also draw some similarities there. So when I said about the complement, the kind of base pairing that they're complementary, that these are similar and then they don't pair. Instead, A pairs with T, and then guanine pairs with cytosine. And then I talked about the hydrogen bonds. You can see that A pairs with, adenine pairs with thymine in a two hydrogen bonds. And then between guanine and then cytosine, you've got three hydrogen bonds. And then we do have the sugar, the 5-carbon sugar there, as you can see, 1, 2, 3, 4, 5 there. And then you have the phosphate group, and then we have got the nitrogen, the base actually, which is the 4 bases that we're talking about. And then, as you can see, the other thing that's worth noting on this slide, it's about, if you can see, we've got the arrows at both extremes. On my left here, you've got an arrow that points from 5' on top, going down, we've got 3' here. And then on the other end, on top, we've got the 3', down here, we've got 5'. So the DNA structure is said to be anti-parallel. So anti-parallel meaning, number one, they're parallel, but then they flow in different directions. They're never flowing in one direction. And I think it's very important also to note these kind of directions, 5 to 3' end or 3 to 5, because they become so crucial when we start talking about how the genetic information flow from DNA, RNA, and then to proteins. And then, as I said here, in RNA, which is a single strand, uracil has a methyl substituent at position C. So I've just given you another second difference here. The clue is, like, DNA, we've got, it's a double-stranded, and then the RNA is it's a single strand. So I think this is the second difference between DNA and RNA, but we've got a couple of them. And I think this should be a take-home assignment as we go to dive deep into other differences, like how DNA differs from RNA. So having looked at, having appreciated the structure of DNA, we now go to appreciate DNA replication and its significance in biological systems. So the word replicate, we've got two ways there. We've got DNA and then replication. So when you talk about replication, this is a general word that you and me have used in a number of scenarios. When you talk of replicating something, simply means is to generate a copy of something. I think a very good example would be in maybe an academic setting where a lecturer is trying to, he's got some exam to administer, and then he makes a printout of the main examination paper. And then from there, he can make several copies of the same exam paper, maybe 1,000 copies that are exactly the same as the original one. So similarly, in general replication, what happens is we are doing nothing other than producing or generating copies of the same DNA. So it is a biological process. The whole aim is to simply create identical copy of a DNA molecule. This occurs during cell division. You may recall, maybe in high school, you had these terms, mitosis and meiosis, under cell division. And then why is it important? Because it ensures the preservation and passage of genetic information. You will agree with me that cells in our bodies, like, they die now and again. So because they die now and again, they also need to be replaced at a constant rate. Because they're dying at a constant rate, they also need to be replaced at a constant rate. And because of that, DNA replication comes in, to ensure there's preservation, but also the passage of genetic information. The other one is essential for growth, development, but also cellular function. So this is the same DNA, just in a degraded manner a bit than what we've had. I want you to take note of a few things here in regards to replication. So one thing when you talk about replication, we ought to have the original thing that we like to make copies of. They won't be replication if we don't have anything, I mean, in the first place. So if you are to do replication, you must have the original stuff that you'd like to replicate to produce copies of it. So we've got on my left here, we've got this strand that we've labeled new strand, and then we've got template strand. Now, just as it happens in the photocopying or pre-production of copies, the hard copies in the exam setting that I gave, similarly, is also a case in the DNA replication. We need to have a template strand. So this is what is being used to make a new strand. So that's why we have new strand and then we have a template strand there. And then as you can see, the template strand seems to be a finished product because this is original. And then the new strand is something that's being made, as you can see. It's halfway through. And the second thing that you got to note is if you look at the template strand, it runs from 3' end to 5' end. Whilst the new strand, it runs from 5' end to 3' end. And as you can see here at the 3' end, at the OH here, hydroxyl group, this is where the addition of new bases is taking place down. So the direction is from is from 5' end to 3' end. And then we've got the main pair in this process of replication in the name of DNA polymerase. So I think for now, let's take note of these few things, and then I'm going to elaborate more in the next slide. Okay, so this is now you want to appreciate the whole process now of DNA replication, how it happens. So basically, we've got all these processes, 1 up to 6, how the DNA replication takes place. So we are starting with the parental DNA, which is a double-stranded, running from 5' to 3' end. This is a leading strand. And then you've got the lagging strand from 3 to 5. So the anti-parallel nature of DNA, they don't go in the same direction. So basically, before the replication starts, the DNA is sealed, is intact as a double-strand molecule, DNA molecule. So what happens now? The first phase of DNA replication is what we call initiation. So what happens at initiation, you can take note, there are mainly 2 things that happen under initiation stage. Number one is that we've got I think the other thing that you need to be taking notes is about the different kinds of enzymes that are key in these processes. So the very first one, we've got an enzyme that's called a helicase. So what enzyme helicase does is it will open up the DNA molecule, separating these 2 strands. So when that happens, it forms what we call the replication fork. So as you can see, this structure now, the parental DNA, it has been separated on this phase, initiation phase, and then you've got now 2 strands. We've got the leading strand on top, and then you've got the lagging strand below. So when that happens, we've got the second stage, what we call primer synthesis. So primer synthesis simply means the production, synthesis is just production of a primer. So what happens, we've got the second pair of enzyme under this phase, and this we call primase. So what primase does, it will come and then bind at each of these strands, the leading strand and then lagging strand. But I'm just going to focus primarily on leading strand. So the primase will come and then bind at the leading strand. So what primase does is it wants to put up a primer where the DNA, where the actual replication can take place. So what it does, it'll come and then bind at the leading strand. When it binds there, it starts adding in the the base pairs, the 4 base pairs which you saw previously. So the addition is not just doesn't just happen in a way. It happens complementary to the information, to the bases that we have on the DNA molecule, parental DNA molecule. So what it means, if you've got a G there, the primase will add a C to pair with a G. If you've got an maybe adenine, it will add thymine. So that's how it does. To make sure it preserves the complementarity nature of the DNA molecule. So this is just a short strand, what we call a primer. So when it adds that, it disengages, and then DNA polymerase now, this is a third enzyme, comes into play now. So DNA polymerase will come and then we'll start adding the 4 base pairs we talked about from the primer, going in the direction of 5' to 3' now. So what you see there, it will be now, we call it elongation. It will start elongating the new strand that's being formed now by simply addition of these bases. But the addition, as I said, doesn't just happen. It will be happening in terms of what's on the parental DNA. If you have a G, it will add a C. If you have a T, it will add A, like that, to make sure they're complementary. But the direction that it will take, it will be from 5' to 3' end. And the other thing that's worth noting in this stage is that the DNA polymerase only adds these 4 bases in one direction, 5' to 3' end. It doesn't, it never adds them in reverse, like from 3' end to 5' end. So that brings us to a different way of addition of bases on this striking strand. Now, because we've talked about the opposite direction of the flow of information, so you can see on the lagging strand, we've got 5' end and then 3' end. So it means if you are to start from adding from this far, we'll be going against the order of how DNA polymerase does this thing. So we can't add from 3' end to 5' end. It won't be possible. So what it does now, it starts to add the bases in fragments. So the primase will come along the parental DNA in reverse. It will put up a primer, as I explained earlier on. When it puts up a primer, and then the DNA polymerase comes in and then start adding the base pairs from reverse order now. It won't be straight. So instead of adding from this direction, going that direction, it will now start add from this end, going this direction. It will be the opposite of what was happening on this reading strand. So this is how it will be adding, just to make sure that it doesn't violate the rule that the addition should always be should always flow from 5' end to 3' end. And because it adds in fragments, and these fragments are called the Okazaki fragments. So when the addition of these Okazaki fragments, when it's completed, now those primers that chopped off, they're removed now from the strand, from the strand. They're chopped and then they're removed, and then DNA polymerase will come and then put in the base pairs to make it complete. When that happens, we've got another step, what we call proofreading. So there could be mistakes during this process. So proofreading comes in just to make sure it collects all those mistakes in terms of where maybe you're supposed to pick a C, you had put a different letter. So proofreading is done to make sure that the replication has been made in a way that leads to DNA molecules, different DNA molecules that have been replicated efficiently without any challenge of that sort. From there, the last but one, we've got what we call ligation. So what ligation does under this step, we've got another enzyme there, which we call DNA ligase. So what the DNA ligase does is it moves now along the both DNA molecules that have just been made. So it will go along the strand to seal the bond kind of strengthen whatever we've been adding in a complementary form. So it will move along the strand, sealing or strengthening the bond. When that happens, then we have the velar strand termination. So by the end of it, we should be able to finish up with two separate rather, not different, two separate DNA molecules that have been generated from a single parental DNA. The other thing that's worth noting is because we've used the parental strand to generate another strand, so the DNA replication is said to be same conservative. Why? Because we've used the old parental DNA strand to generate another strand to make one complete DNA molecule. So in short, this is how the DNA replication takes place. And I have a short video, less than 2 minutes, just to sum up what I've said. So I just play it so that it just sums up whatever I've said about the whole process. So kindly listen and take notes. Sorry, I just have to confirm with me, is the video audible?
I can see it. Haven't heard anything yet.
Sorry?
I haven't heard anything
Is
yet. Should it has any sound?
Yeah, it has to produce sound.
I didn't hear anything yet, but maybe it hasn't talked yet.
Okay.
I think there is no voice coming in. Maybe like you may have to like click something on the microphone to let it let the system play the sound as well. I think it's playing on your computer speaker right now, and the voice is not coming through.
So there's no sound.
Yeah, I can't hear anything from the video.
Okay, that's okay. So I think, as I said, I think I'm just gonna pause and then just cover it with some explanation, little quick. So as I said, this is the DNA helicase, as I said. So what it does here, it will just kind of separate the two DNA strands, and then it creates what they call replication fork. I think this is what we see now. So this is a DNA helicase. This is the double strand, and then when it comes, this is the initiation, and it kind of separates the DNA molecule into two strands. You've got the leading strand up there. You've got the lagging strand down there. So second now, this is second enzyme primase that has come up now. What would you do? This is primer synthesis. It will just come and then put up a primer, adding some new nucleotide bases to form a primer where the DNA elongation can start from. And then we've got the DNA polymerase binding at the primer to start elongating now the chain. So I think the other thing to note here is that the direction is from 5' to 3' end, and then the bases that are being added, they're complementary, as you can see. So this is now the lagging strand where I said because it's not learning the direction of 5 to 3' end, it has to do in reverse now. So the primers will start adding put up a primer in reverse order, and then the addition of the nucleotide will also be in reverse. And then you will just, at the end of the day, concatenate all the segments to make a complete strand. So the after the whole thing, the primase now removes the primers, and then the DNA polymerase will come and then seals in those gaps with the root stuff, the required nucleated bases to form a complete strand. So what you see here, this is a DNA ligase just setting up the newly formed DNA molecules to strengthen them. Yeah, so that's about the DNA replication. Just sort of about the sound that you cannot hear the sound, but I think when we share the slides, you should be able to hear the sound, and that will even make it better and easier for you to follow through. Basically, that's what replication is all about. So, having talked about DNA replication now, we now want to appreciate now how this, after the information has been replicated now, we want to appreciate how this information moves from the DNA to RNA in the process of transcription, from RNA to amino acids in the process called translation, and then after that, the amino acids undergo some crazy folding that will eventually give us a protein. So when you talk about transcription, this is, in simple terms, is the process by which the genetic information that is encoded in the DNA is copied into a complementary RNA molecule. Get this one, complementary RNA molecule. So it's not just adding, it has to take note of that. And then, unlike in DNA, where we're using the DNA polymerase, this time around would be the key player is RNA polymerase. And require it, and for it to perform the functions, it requires the DNA template, activated precursors of the four ribonucleotides. We've got A, C, G, and then U. Because now we're doing RNA now, we're not going to be having a U, a T. Instead, we're going to proceed with uracil. we're going to see in the subsequent slides, and then some ions also. So we've got three major steps that happen during the transcription. I think because of time, I will just go straight, and then I will make my explanation based on the. I also have a small animation, so I think I can use that to explain the how these steps are important, and then how we can go about them. But then, I just want to explain a few things here. We've got the DNA molecule. We've got two strands. We've got the upper strand in red. We've got the DNA strand in blue. And then we've got the RNA polymerase. And the direction is from 5. Um, the direction of the DNA upper strand is from 5 to 3 prime end. The direction of the DNA lower strand is 3 to 5. So it means that gives us a clue, or gives you a clue. If you're to transcribe either of these sequences, you've got to be mindful of the type, the kind of direction that you're going to flow based on the template that you're working with. So assuming I was working with the down strand, the one in blue, because if this is a 5, this is 3, and then if I'm to do my transcription in 5 to 3, obviously I can't start from here, because if I start from here, it will be from 3 to 5. I have to start my transcription from 5 to 3. So what basically happens also is that I know there's a lot of chemistry that is involved. Along the DNA strand, you've got what we call the promoter region. So the promoter region, this is the region that signals the start of this process, the transcription. So the promoter region has got with it the what we call the transcription factors. I think there's a lot of chemistry involved. I'm not just gonna go those details, but one thing to note that there is always that promoter region that signals, you can take note of that, that signals the start site of transcription. So basically when transcription is taking place, the major thing that happens also is the addition of these bases that will be complementary to what we have on the DNA template here. So as I said earlier on, that in DNA, we've got a T, which is thymine, but when we start dealing with RNA, we no longer deal with thymine. Instead, we deal with uracil. So the RNA sequence here in green, this is being transcribed from DNA from the lower strand as a template strand. So when you have the A, the RNA polymerase will add a U here, not a T, because this is RNA. We've got a C there, the RNA polymerase is going to add a G complementary. And then like so on and so forth, AAT like that. So that's how the addition of the 4 nucleotide base pairs is going to be added. I think you're going to appreciate more when you have the animation. And then so basically here what happens is the DNA molecule also has to open up for transcription to be possible. So the binding of the RNA polymerase at the promoter region leads to the opening up of the DNA molecule. So when that happens, then RNA will start doing its job of adding the nucleotide bases, what I said on the previous slide. And down here, what we see is the complete RNA that has been transcribed or that has been made from the this DNA molecule. So in transcription, we only use a single strand as a template because we're also producing a single strand. So as I said, the second difference between RNA and DNA is that the RNA always is existing as a single strand. As you can see, this is the RNA. It's never a double-stranded. So because it exists as a single strand, we're not going to need the both strands. We only also need a single strand to do the transcription. So I'll also in 2 minutes use this animation for us to follow through easily. So as I said now, we'll talk about transcription. For it to start, we need to have a promoter region, something worth taking note of. It shall always start at the promoter region. So when you read deep, you find that there is, as I said, what they call transcription factors here, and all what they do is to signal the start of transcription. And what we have there coming there, this is an enzyme, RNA polymerase, and then it will come and then bind at this region. So I think the other thing now, as people that have got growing interest in synthetic biology, this is a crucial stage now because this is where most of gene expression is controlled. You can choose to play around with this process, with this stage where to block the transcription, or to do some other kind of modifications. But this is the critical stage of gene expression. So you got to take note of this particular stage of phase. So unlike the DNA replication, where we've got the DNA helicase, in this process, we want to have the RNA polymerase. And then when that RNA polymerase binds the promoter region, it will cause the DNA molecule to open up. When that opens up, then the primer now will be synthesized. The primer will enable the RNA polymerase now to start adding the base pairs as we saw in replication also, but in 5 to 3 prime direction. That should always be taken note of. So basically, but I think that's the whole process. As you saw, RNA um that has been transcribed as it was happening. The chain kept growing bigger, kept growing longer rather. So this is how the whole RNA is made as regards to transcription. The very last thing to note is that the after starting at promoter region, the transcription when it reaches the terminator region, that concludes the whole process of um, transcription. When that happens, then the RNA polymerase disengages, dissociates from the DNA molecule. The RNA that has just been made leaves also, and it undergoes what we call some process that we call the post modification, getting it ready for translation. So I think some of the things that we need to take note of, what happens that when the RNA is made, it has got with it two types of regions. It has got what we call the coding regions and then non-coding regions. So the coding regions are what we call the exons, and then the non-coding regions are what we call the introns. And then for us to have a RNA that is ready for translation, we've got to get rid of those non-coding regions. I'm just gonna play and then show how this is done. So what we see here is actually the spisosome, an enzyme, is getting rid of uh, the introns, the non-coding regions that we do not need, getting the RNA ready for translation now. So the whole process of transcription happens inside the nucleus. When all is done, the messenger RNA go to leave the nucleus through a nuclear pore to cytoplasm, where the translation takes place. So this marks the end of what transcription is all about. So the process has been transferred from the DNA to now RNA, specifically the messenger RNA. Now we want to talk about the last step in the central dogma of molecular biology, how the information is transferred. So after having the messenger RNA or the RNA that we've just made, then we've got to go to the very last one, which is called translation. And now we cannot talk about translation without necessarily talking about the genetic code. Now, when we talk about genetic code, it's not some jargon or some big words, some big word rather. The genetic code, I mean, our friends that are coming from computer background, maybe maths background, those that have done, or those that do programming, be it Python, R, or what have you. You know, we do also have these codes in programming, and what code is simply a combination of numbers, sometimes numbers and then sometimes symbols that represent a particular thing. So in biology also, we do have this genetic code, which is a combination of letters that simply represents a particular amino acid or particular protein. So when you hear about genetic code, it's simply the combination of those letters standing for representing a particular amino acid. So genetic code is just relation between the sequences of bases of DNA or mRNA and the sequence of amino acids in proteins, as I said. So what happens? We've got the three nucleotides that encode amino acid. So because we know where we're coming from, that these nucleotides have been represented by letters, as we saw. So those three, when they come together, they can represent or they encode for one amino acid. So basically, the basically it's 20 set of amino acids, and we only have 4 bases. So simple calculation shows that a minimum of 3 bases is required to encode at least the 20 amino acids. So one thing worth noting is that a group of 3 bases is called a codon. We're going to use from henceforth, we're going to be using this term codon. The genetic code is said to be non-overlapping. So what that means is, if considering that we've got this sequence, we've got A, B through to E, A, B, C, D, E, F. So in an overlapping code, what would have is, because they overlap, we would start with A, B, C, and then instead of us counting from D, because there's that overlapping nature, I would start from B, C, D, like what we have here. And then the other code would start from C, D, E, F, like that. So this is a scenario where overlapping nature means. But then in an overlapping, the same sequence would be read in this way. Would start from A, B, C, and then from there would jump and then start from D, E, F, like that. So the genetic code, how we read it, how it's read, is read in non-overlapping manner. So with me here on this slide, we just have the genetic code. Quickly, we've got, it's simple to read. I'm just going to demonstrate how this is read. We've got letters here, a group of letters. three, which I said these are codons. And then we've got the corresponding amino acids. So basically the 20 amino acids, as I said. And then how this is read, it's quite simple. You've got here to my left, first letter. Up there, we've got second letter, and then on my right here, we've got the third letter. So I'm going to start demonstrating here on the very first column. We've got triple U. So the first U, you're going to take it from my left, which is this U. There, you're going to take it there. Second U, this comes from above. So you're going to take this U. This is our U. And then the third U, this is what we see. This is the U, the uracil. So what it means the three letter code U encode an amino acid in the name of phenylalanine, which has just been written in its short form as phe. And then secondly, you've got UUC. Again, they took a U from this side, U above, and then U from the other side. And then the last demonstration, I'll do it down here. We've got GAU. So the G comes from there, my light, and then A comes from above, and then U comes from my left. And then we've got GAU. And then this codes for asparagine. So the other thing to note is it's possible to have more than one codons encoding for one amino acid. As you can see here, serine is got all these 1, 2, 3, 4, like 5 codons coding for one amino acid. So it's possible, like one amino acid to be encoded by more than one three letter codon. So I think something to take care of, to take note of. And then what I have here is the same one, just the full names of the amino acids, but the language remains the same, what I explained. But so you've got all the 20 amino acids, and then you've got these three. These are what you call the stop codons. So we're going to see what role they play in translation. So we've got 64, the combination 64 combination, but then out of 64, three of these codons are referred to as stop codons because they signal the end of translation during the process. I think I'm going to demonstrate as we start looking at translation. So this is something that you need to keep so close to you and know it by heart so that it becomes so easy for you when you start to play with these sequences, how you maneuver from DNA sequences to protein sequence or to amino acid sequence. So I think we've got a few things that we just have to know about the genetic code. Number one is code no punctuation. You just read as it is, a 3-letter word code rather. And then the genetic code is degenerate. What we mean is we've got some amino acids, as I explained, that are encoded by more than one codon, since there are 64 possible codons and only 20 amino acids. So there's that possibility that one amino acid could be encoded by more than one codon. So in fact, 61 of the possible codons specify particular amino acids, and 3 codons, which are stop codons, designate the termination, as I said, of translation. The last one to note is that the genetic code is nearly universal. So like this is true for about 90% of organisms, like political systems that are there in the world. So we're going to move to look at protein synthesis now. So the importance of the genetic information lies in the proteins that it encodes. So I think the whole where we're coming from, it was just to help us to know how we can maneuver, how we can read from DNA to RNA that without without reaching this far, all that information won't be helpful, because by the end of the day, all we need to know is that whatever we have under the genetic sequence, a DNA sequence or RNA sequences, how much of that information is made available, is translated to to the phenotypes, something that we can see. You've talked about different genes having different functions. I mean, all that information can be well understood when we reach at this stage where we want to produce proteins based on the information what we have gotten from the messenger RNA. So that's the proteins play most of the functional roles in cells, and this process takes place in liposomes, unlike other processes that uh, take place in the inside nucleus. So we've got another player here, the transfer RNA, messenger RNA, and then many proteins participate in this process. I think you're going to see in a few minutes to come. So the protein is synthesized in the amino to carboxylic direction by the sequence addition of amino acids to the carboxyl end. So I think forget about the I mean the wording there. In simple terms, we are going to witness what we've been witnessing transcription, the simple addition of um things along the chain. Only that this time around we won't be adding in the nucleotide bases. We're going to be adding in the amino acid, and the direction remains the same, but what we are saying now, what matters, where are we going to be adding these amino acids as translation takes place? I think this is what we're going to appreciate. But as I said, um, the fidelity of protein synthesis requires the accurate recognition of three bases, codons on messenger RNA. I think this is another important statement, which says the fidelity of protein synthesis requires the accurate recognition of three bases, codons on messenger RNA. Or as you might have seen from the genetic code, if you have a three-letter word, any replacement or substitution of a single letter may result in two different amino acid. So, if, for example, if you have maybe U, and then you're going to substitute maybe two of the U's, and then you put maybe GG, you put the different bases all together, you can end up um having a different amino acid being expressed. So I think a lot of care has to be taken in this regard. So I think I want to explain to you how the whole process is how the whole process takes place. So the main player now in this scenario, in this process, we've got what we call the transfer RNA, which is basically just written as tRNA, a small t. So the small t, it has to do this based on the information that's available on the messenger RNA. So the messenger RNA, as we see it here, it's got the those bases, four bases. You've got the GU along the whole strand. So what will happen is that the transfer RNA, tRNA, will come with it the amino acid. So this is the attachment site up there where the amino acid attaches at this transfer RNA. But then the reading is done down here. So the transfer RNA comes with it what we call the anticodon. So the anticodon at the base of it, it carries with it these bases also that are complementary to the messenger RNA. So when it moves, it will start now reading now. So it will come in three, because we know we are dealing now with the three letter word codon, which encodes for a particular amino acid. So for example, if you've got serine up here, so what we'll have down here, it will be a three-letter code that is responsible, or that encodes an amino acid cell up there. So it's going to come and then read here. When it reads, it will disassociate, and then the amino acid here, it also detaches and then starts forming an amino acid chain. I think I'm going to explain better. Also, you're going to appreciate better when we start looking at the minor animation that I have with me on this slide, on the presentation rather. So I think this is something broad. So it's the same translation, just in the linear presentation. So what I was showing you, the attachment side is just the other way, the upside down. We had it previously, it was on top. But what happens? So the anticodon, the tRNA, you've got the attachment side. So you've got these kinds of amino acids, but each one of those have got the 3-letter code specific to them. So during the addition, as I said, it'll be what we have here will determine what's gonna be read down there on the messenger RNA. So the reading frame, I think that's another point that we got to take note of, is determined by selecting the insertion codon. Typically, this is the start codon. It's AUG. So this is where the translation takes place. AUG is the start codon. It signals the starting point of translation. And then I also showed you the other stop codons where they will signal the end or the termination of translation. So a bit of chemistry here, but not much. So this is the messenger RNA, the letters as usual, as you see, the bases rather. And then you've got the transfer tRNA here, and then you've got the attachment site, the attachment site where the amino acid attaches. So when they come, they will lead. So in a complementary way, as I said. So as you can see there, the 3-letter codon, C pairs with G there, U pairs with A, C pairs with G. When it reads like that, then the amino acid exits through this point. So you've got the attachment side, you've got here where it reads, and then it exits there. So when it exits, every time it comes, it attaches, it reads, and then it exits. It forms the chain of amino acids. So as it happens, the amino acid will just be added to this going chain, a polypeptide chain of amino acids, until it reaches a stop codon. When that happens, then it will just disassociate from the messenger RNA, and then we shall have left with the chain of amino acid. So when what we call here, this is called the liposomal. It's a liposomal small unit. The whole thing here, this is a larger subunit. These actually they bind, and when they bind, this is where the transfer RNA sits in, and then they move like that along the messenger RNA, adding reading the information on the messenger RNA, and then producing the appropriate amino acid. So the direction, I think one thing to note here is about direction. It remains 5 to 3 prime, but I think everything is as what I explained in the previous slide, just that it's more of a decorated presentation, but it's more of what we said. So I talked about the large liposomal subunit, smaller liposomal subunit, and then you can see inside the transfer RNA bringing in codons to read the information on the messenger RNA. So this is a small 2-minute animation just to wrap up what I've said as concerning the process of translation. So we are starting from this point now. We've made the messenger RNA that moves from the nucleus through a nuclear pore. So a nuclear pore is just a minor hole, uh, where the messenger RNA goes through from the nucleus into cytoplasm. And you should take note that the translation, the whole process takes place in the cytoplasm. So what we see here is the messenger RNA leaving the nucleus through a nuclear pore to start translation. So what you can see here is actually basically we've got the both the small and then large liposomal subunits binding to the messenger RNA, and then you can see now the transfer tRNA bringing in now the three-letter code now to start reading the information from the messenger RNA. So as you can see, as this happens, what we have here building up, this is the chain of amino acids. So when it comes, it reads and then it disengages, and then the the amino acids chain keeps going because they keep being added on this growing chain. So I mean this chain now for amino acids, after everything, we now have the complete chain of amino acids. Now for the amino acids now to become proteins, now as I said, they undergo some crazy folding. So it is that folding that now gives us what we call now proteins. But one thing I would like us to note here is that so the information now has been read from the DNA to RNA. Now we have proteins now. So when we hear people talking in layman's language to say we would like to isolate genes responsible for virulence, genes responsible for drought resistance in plants, maybe genes responsible for A, B, C, and stuff like that. Basically, they go through the whole process that we've undergone. DNA, RNA, and then proteins. So what we see outside is actually the proteins now. When we see when we say this resembles that in terms of maybe parent and offsprings, we can only we don't see the magic at the genetic level. We don't see the magic at RNA level. We can only see the magic at the proteins level. But now this is so I think that's a bit of this knowledge that for somebody who is interested in synthetic biology will be able now to make adjustments or to make modifications at the genetic level that would manifest in the phenotype, what we see outside. So the folding has taken place. We now have ended with proteins now. So this is basically where the central dogma of molecular biology ends. Is that and that ends my presentation. But before it ends, there's that question that we need to take home with us. And the question is, so Heidi talked about what synthetic biology is all about. We've now come to the understanding, to the knowledge of central dogma of molecular biology. Now, by now, I should be able now to bridge the gap to say, so how does the central dogma power synthetic biology? I mean, if I've got a growing interest in synthetic biology, how do I use this knowledge now? I've known how the information is read, is transferred, and then how proteins are made. How do I use that maybe to contribute to the medical research, be it drug discovery, maybe in agriculture, I mean modifying genes for drought tolerance, or I mean stuff like that. So that's the beauty of it. So I want each one of us as we go to have this as a take home, and probably this should be able to open up to start imagining the countless possibilities that synthetic biology has as we embrace the future. The very last slide says science ceases to be such the moment we stop asking questions. I want to conclude here, but before we start taking questions, I want to be advised by ICE whether we've got enough time for the questions. If we do not have enough time, we always have the Discord channel where we've been interacting from last week, and we can always use that for interactions, and be assured that you also have this presentation for just to I mean to blend in where you may have missed a thing or so. So thanks everyone, and I want Ice, you can advise whether what do we do from now.
Yeah. So I think we are almost at the time that we planned. So like if you guys like, you know, in the attendees have to, you know, do some other things, please feel free to leave. But let's take maybe like 5 or 10 minutes here, just in case anyone has any question, and then we can chat a bit. And then everyone, you can access like this recording, and we will try to like upload it in a week. And the other thing that I would like to say is that like we have the homework associated with this lecture. So I'll post that in the chat, and also put that in our email and Discord, just so that you can access them. Hey, Kado, question?
Okay. Ice, maybe I would like to talk about something. I don't know.
Wait.
Ice, can you hear me?
I can hear you. Yeah.
I would like maybe to talk about something. I think maybe we have made students stay much more time here. Maybe for the issue of questions, people who have much more questions, they can send the questions via Discord, so that they can get much better feedback. Because I don't think we should keep students much more longer. Maybe like I had some small video which I wanted everyone maybe to get a look at. It's talking about synthetic biology. It's very short, and I know it will be much more exciting. I don't know if the instructor for today is accepting.
Yeah, like putting things in Discord would be perfect, I think, because like we have some record. Just so you know, like, you know, most of the chat in the Zoom is pretty hard to record and pretty hard to like you know, share. So like if you guys like, you know, like I think feel free to ask like some simple question along the lecture, and if we see them, then we'll respond to them. But like something that would be beneficial for everyone, we can put that in Discord and we can talk about that for sure.
Okay. Thank you so much, Ice. I request maybe Steve to first stop sharing so that I can pass on this video. It's a very short video.
Can you say that again?
I was saying, like, I request Steve to stop sharing. And
Oh, I see. Okay.
Yeah.
Thanks a lot, Steve. Water, you have a question, right?
Yes, I basically have two questions, and this is coming from the section of DNA replication and transcription. So when you look at replication, we realize that the strands are separated, and then we have new strands being synthesized, and afterwards they get back together to form two new strands. But when it comes to transcription, we realize that the messenger RNA, as it is being synthesized, the fact that it is complementary to the template strand, would expect it to anneal or to bind back to the template strand, but it doesn't do so. It gets actually off. So I was wondering what binds it or what stops it from binding back to the template strand, and yet it is complementary. Then secondly, still I would be looking at that I'm trying to forget that. Maybe as you answer the first question, I will get back the other. Thank you.
Yeah. So Steve, you wanna talk a bit? Cool. Yeah. Please go ahead.
Okay. Okay. Thanks. So the point is, why does it not bind back to the main strand? I think basically one thing that's worth noting is that the whole purpose of why we have the messenger RNA produced is that the flow of information keeps going. So we don't want to have it stuck in the binding to one of those strands. We want it straight out to go into the cytoplasm where the translation will take place. So basically, as soon as it finishes, then through relevant signaling, it leaves the nucleus, and then straight away it goes into the cytoplasm to kickstart the whole process of translation. I don't know, Ice, you are free to add in if you have something.
I think that sounds good. Kato, Water, you want to ask the second question first, or like maybe Kato can share the video that we're looking at, and just in case, like if you guys can stick around a little bit after the question, maybe we can take a group photo real quick, just so that like we know that we all attend here. There are a lot of people attending this lecture, and we'd love to like just take a quick picture that we are here all together.
Okay, I think Ice, with your permission, I see we've reached 8:30. I think it's the time that we're supposed to be out of here. I am afraid maybe we could let Ima do a video, and then we take care of all the questions in the Discord. I'm not so sure if that works for everyone, but yeah.
Okay, sure. Yeah, maybe Water, if you have like the other question, if you can type it into the Discord, that would be great.
Yeah, I think let the questions go to the Discords, and we leave people to go. But before maybe we shall take a group photo, if at all, for those ones who are in position of maybe. So I will play this video. Maybe it's a very short video.
We could design life the way we design gadgets and.
I don't know. If
Imagine a world where we could.
I can hear it.
Is it?
Yeah, we can hear you.
Okay.
Yes, Ima, go ahead, please.
Imagine a world where we could design life the way we design gadgets and machines. Sounds like science fiction, doesn't it? But this is the revolutionary world of synthetic biology, where scientists combine biology and engineering to create new forms of life and redesign existing ones. So what is synthetic biology? At its core, it's about understanding and redesigning the fundamental building blocks of life, DNA. Each organism carries unique DNA sequences that determine its characteristics. Synthetic biology takes this concept a step further. It manipulates these sequences to create organisms with new desirable traits. You might be wondering, how does this work exactly? Let's take a journey into the microscopic world. Picture a long ladder twisted into a spiral. This is DNA. Each rung of the ladder is a pair of molecules or base pairs. These pairs form the genetic code that instructs cells how to function. Synthetic biologists can change these instructions by adding, removing, or rearranging these base pairs. This is like rewriting the software of life. Now, this might sound a bit like genetic engineering, and you're not wrong, but synthetic biology goes a step further. Whereas genetic engineering often involves modifying a few genes, synthetic biology aims to construct entirely new genetic sequences, effectively building new forms of life from scratch. The potential applications of synthetic biology are vast and varied. For instance, in healthcare, synthetic biology could engineer bacteria that produce life-saving drugs. In agriculture, it could create crops that are more resistant to disease and harsh weather conditions. And in energy, it could develop microbes that generate biofuels. However, like any powerful technology, synthetic biology comes with its share of challenges and ethical considerations. The possibility of creating new life forms raises questions about safety, biosecurity, and the implications of playing God. It's a field that needs careful regulation to ensure its benefits are harnessed responsibly and ethically. So to recap, synthetic biology is a revolutionary field that combines biology and engineering to create and redesign life. It works by manipulating the DNA that form an organism's genetic code. The potential applications are vast, spanning healthcare, agriculture, energy, and more. However, the field also raises ethical and safety considerations that need careful thought and regulation. Synthetic biology is a testament to human ingenuity and the power of science. It's a field that's to transform our world in ways we can only begin to imagine. So as we stand at the threshold of this new era, it's important for us to understand, engage with, and shape the direction of this transformative science.
Okay, that's what I wanted to share, and I hope it will help add on the lecture for Heidi of last week and for Steve of this week. Thank you so much.
So before we leave, let's take a minute and like maybe take a group photo. If any of you would like to open up a camera, that would be great. But if you don't, then that's also fine. I'll just like do the screen cap of like people showing up in Zoom. But yeah, if you have the camera on, I'll share this picture on Discord.
I think IC will take the picture, right?
Yeah, I'll take it, or you can take it as well. I'm doing the screenshot.
You take it.
Okay.
Okay.
Perfect.
Let's all rather show our videos if we are interested in the picture.
I'm taking the first one now. I'll keep taking it.
Yeah, let's turn on our videos for those ones who want to be in the picture. Let's turn on our videos and we create this wonderful memory. As we are taking the picture, I wish to inform you that one of the teams we have been mentoring, it went to France for a synthetic biology competition in Paris, and this team won a silver medal. So we hope to have many more teams from Africa we shall be mentoring to participate in synthetic biology competitions. And we ensure the mentorship we shall be providing, it will help in developing both of the students and the teams at large. And we shall get much more startup companies in synthetic biology through SynBio4ALL Africa. And we are going to start on building a synthetic biology community. So we hope you will join that community and be part of the new era, which is trying to use synthetic biology to solve the global problems. Yeah. I appreciate everyone who has joined in, and I request even you call on those ones who haven't joined in to come and join in. This course is real an expensive course if you decided to pay for it, but we have decided to bring it to you at the free cost. So please invite much more of your friends to come and join in. And I encourage you to do the homeworks which the instructors are giving and to continue engaging with us. If at all you need something, you can contact us via emails. We have our email SynBio4ALL@gmail.com. You can contact us. We are creating
Imagine a world where we could design life the way we design gadgets.
Okay, cool. I got a
Yeah,
lot of pictures, and I'll share with everyone.
Thanks.
So thank you everyone for showing up.
Thanks.
Have a good night.
Thanks a lot, guys. Thanks. No, thanks a lot.
Thank you everyone for being here.
Yeah, I wish
Thanks,
you.
bye, and we remain. Yeah, we remain SynBio4ALL and goodbye.
Good night, everyone.
You just reach out to us if at all you have any question. We shall always be there to answer you. If you want to form a synthetic biology team, please reach out to us. We shall help you. Thank you so much.
Bye.